Okay, so a very good evening to one and all present here. Welcome to the course on Introduction to Complex Biological Systems. I am Srijan Sen from IIT Kharagpur, and today we'll be taking the week 5 live class session. Okay, so yeah, so let us, so I'm sharing my slides with you all. Uh, yeah, I guess my screen is visible to you all. Fine, okay. So today we will basically talk about cells. So cells are an important uh, unit of life. So we will understand about quite a few basic and advanced level of understanding on the cells. Okay, so let us start. So we know that there are three domains of life, starting with bacteria, archaea, which falls along the in the category of prokaryotes, and among the eukaryotes,
Sir,
yeah,
sir, not able to hear your voice, sir.
not able to hear my voice. Uh, am I audible? Am I audible? Okay, thanks. So basically, there are three domains of life where we have the prokaryotes, uh, like the bacteria, the archaea class, and then we have, say, the eukaryotes, where you have human, yeast, and then there are different categories of all of that. Okay, now. So all cells are either prokaryotic or eukaryotic in nature. So prokaryote means like they are single celled organisms, and all of them are bacteria. And eukaryotes, they are single celled or multicellular organisms. So pro means before nucleus, and eu means it has a true nucleus that is present. Now if you see the prokaryotic cell versus an eukaryotic cell, you will find multiple differences, but even if we're not going into the differences, let's just think of the similarities of what they have, and you would see that they have a plasma membrane. You can see find the cytoplasm, the genetic material, which is the DNA, the energy currency in the form of ATP, enzymes, and the coenzymes. That's the mostly the internal organization of the cells. Now, So the cell therapy, what it says, it's that all organisms are made up of cells. So the cell is the basic unit of life in all the living things, and all cells come from the existing cells. And this is very important because it shows that all living things share a similar structure. Now, there are two types of cells, like we discussed, it's a prokaryotic and an eukaryotic cell. So, all cells, whether they are prokaryotic or eukaryotic, have some similar features. Like you can see the plasma membrane, then cytoplasm, like we discussed before. So, there are certain things which are similar among them. Now, organelles are structures that enables the cells to live, reproduce, and grow. So there are different types of cell organelles that carry out different functions and are very important for proper maintenance of these cells, and each have different functions. So, okay, so prokaryotic cells, what are they? They have no membrane covering the nucleus. They have no membrane that they have covered organelles. They have a circular DNA, which is also known as plasmids in bacteria. Okay, and you have the eukaryotes that have a proper nucleus. They have a membrane that covers the nuclear organelles. They have linear DNA, and all are under all other cells. So cell membrane is basically the gate into the city, right? So it's the outer layer of the cell. It allows the nutrients into the cell and the wastes outside of the cell. So the cytoplasm is basically a jelly-like fluid that's contained inside the cell that holds the organelles. The nucleus is basically the most important part that has the DNA material. It's the control center of the cell. It has the it has the cell's DNA, and it keeps that. So it's basically the office from where all the functions actually basically happens. Now, mitochondria is the powerhouse of the cell. It provides the energy for the cells to move, divide, carry out the different functions, then development, homeostasis, things like that. Means proper maintenance of the cells are all required, and that is done basically by the mitochondria. So it's the electric company of the cell. Next, coming to the ribosomes, that's the site where the proteins are made. So you know that from DNA we go on to the RNA, and from there is translation into proteins. So it's like that. So the ribosomes is the site where the proteins are being synthesized, and then it moves either out of the cell, or goes to the cytoplasm, things like that. Okay, so the cell parts are made of proteins, and that is why synthesis from the ribosomes is quite very important. So it's the factory where the generation of proteins basically happens. The other part is the endoplasmic reticulum. That's the transport system of the cell. And why is it important is that it provides the roadway of the cell. It maintains the roadways of the cell. Now you have a rough endoplasmic reticulum and a smooth endoplasmic reticulum. So the rough endoplasmic reticulums have the ribosomes attached to it, and the smooth endoplasmic reticulum, there is no ribosome. Okay, now Golgi complex is the packaging house of the cell. So it packages, processes, and shifts out of the stuff that the cell makes. So like in computers you have, in laptops in computers you have a UPS. So it's the UPS of the cell. The other important part is basically the lysozymes, which digest the food particles. Okay, and the cell parts, which is the garbage men, and it protects the cell by digesting the foreign invaders. So it's basically the police. Now vacuoles. So vacuoles. So plant cells have big vacuoles. Most of you know that, and animal cells have smaller vacuoles. So vacuoles is the largest organelle in the plant cell. It stores the food, waters, and the wastes. And coming to the cell wall, so plant cells have cell wall, animal cells do not have a cell wall. So plant cells have an extra layer of protective covering in the form of this cell wall, but animal cells don't require a cell wall, so there is no kind of a protection, things like that. Now, chloroplast. Chloroplast is another organelle, and basically it's mostly found in the plant cells. It contains the chlorophyll that makes the plants green. It's the place where photosynthesis takes place. Okay, and okay. So now, plant cell or animal cell may be a kind of so found in the plant and animal cells. So, like you have the nucleus, the Golgi complex, the mitochondria, uh, lysosomes, the endoplasmic reticulum, the cell membrane, ribosomes, and the vacuoles. So, these are present in both the plant and animal cells. Whereas, on the other hand, in the chloroplast, you find the extra the chloroplast and the cell wall. Right, if that is so, then okay. So the bacterial cell wall, so it's different, it's not very different from the plant cell wall. Okay, so it's similar to that of the plant cell wall. The function is basically quite different. So it lies lies outside the cell membrane in nearly all the bacteria except the mycoplasma and some archaebacteria. Just a minute. So, okay. So the bacterial cell wall, which lies outside the cell membrane in nearly all bacteria except the mycoplasma and some archaebacteria. So there are two important functions, like it maintains the characteristic shape, it prevents osmotic lysis, right? And so the cell wall looks somewhat like this. Okay. Now, okay. So, right. So now we will get into the lipid bilayer. So the lipid bilayer is basically the most important part of the constituent of the cell wall. And so you will see that, so basically you see that it has an hydrophobic head group and hydrophobic tail group. So basically what happens over here is that you have choline, phosphate, glycerol. So it's Um, mainly forms the hydrophilic head group, okay, and then the hydrophobic tails are basically the ones that comprise the inside of this. So it's a phospholipid, so it's a PLP bilayer, okay, where you have the protein, then the lipids, and then the set of proteins. So you have a phosphatidyl ethanolamine, where you have the different groups. So there are different groups that are attached over here. So based on this, based on the whatever the hydrophilic heads are, based on that you have the distinguished things, starting from the phosphatidyl ethanolamine, then phosphatidyl serine, phosphatidyl choline, sphingomyelin, sphingosine, things like that. So the lipid structure of archaea is basically you have a fatty acid and ester bond, and then the glycerol 3 phosphate. You have another one where you have in archaea, it's basically you have the glycerol 1 phosphate attached to by the ester bond, and then the isoprenoid. Okay, so that's the difference in the structure of an archaea and a bacterium, right? So the lipid bilayer, so this is the shape of the molecule, and it's like packing of the water molecules over here. You have the micelle formation and the lipid bilayer. So energetically unfavorable, so planar phospholipid bilayer with edges exposed to water. The sealed compartment formed by the phospholipid bilayer, it's energetically favorable. So the levels of organization of life, where you see there are different polar head groups, then a rigid steroid ring structure and a non-polar hydrocarbon tail. So what you mostly require is basically how these structures look. So you have polar head groups and non-polar tail region. So there is cholesterol strengthened region and the most more fluidic region. So that's the level of organization of life. Now, if you look into this PLP bilayer, what you see is mostly there are different cholesterols in between the in the lipid regions that's packed between this layer. Then you have the transmembrane glycoprotein. Okay, you have some oligosaccharide linkers, the GPI anchored proteins, the glycolipids. So those are actually the different types of membrane proteins that are present. A part of it is in the cytoplasm, the other part is towards the extracellular matrix. Okay, and then a part of that forms the raft domain. Then so everything is basically a compromisation of the comprising of the, you have lipids, then cholesterol. If it's attached to the, if in place of the polar group you have a glycogen group, then it's a glycolipids, so things like that. Okay. Now, the bacterial internal structures, they typically contain, so bacterial cells typically contain the ribosomes, then you have a nucleoid region, the vacuoles, certain bacteria sometimes contain endospores as well. Now, the nuclear is basically the prokaryotic nuclear region. It's located mainly, it's comprised mainly of the DNA, but also contains RNA and proteins. So DNA is usually one large circular chromosome, like Escherichia coli DNA circle measures around 1400 micron. The DNA is coiled extensively with RNA and nuclear proteins. Then you have the plasmids, that's the extra chromosomal pieces of smaller circular DNA. Now, how do you observe the cells? So for that, we use the various types of microscopes. Okay, through eye, we can see up to 1 meter, maybe um, 10 centimeter, or up to a quite few centimeters. We can see up to that. But it's very difficult to see the structures which are lower than that. So you can see a man's height, a hand, a finger, but it's very difficult to understand the thickness of a hair cell and bacterium. For that, you need a light microscope. If you have to see like atoms, molecules, or viruses, macromolecules, then you need an electron microscope. Okay, so there are different types of instruments that are allotted for every particular thing. Now, the light microscope can see up to zero point 0.2 microns. So there are different pieces that are attached. So you have an eyepiece. So you can see over here, you have an eyepiece over here. Then the spring is loaded with clips from where you actually observe. You have the stage where you set it up. You have the different types of objective lenses for zooming into that particular. It's like more or less like a camera of a phone. Okay, then the coarse fine focusing that's required, and the light source through which you would be able to see the it. So it's basically from the eye, it's from the eyepiece you see, it's the objective, the specimen, the light source, and in back it reaches the retina and you're able to see it. It's quite a fast process. So the light path is in the light microscope. Now, electron microscope is basically for the subcellular studies of the different structures. Now you have the scanning electron microscope, which focuses a beam of electrons onto the surface of the sample and provides images that give a 3D representation uh, of the samples. Now, SEM is used to study surface structure of objects. Now, the transmission electron microscope focuses a beam of electrons through the sample. It's used to study the internal structure of the cell. The scanning electron microscope is basically you have an electron gun, then the condenser lens, the beam deflector, you have the objective lens, then the electrons from the specimen are split, and then it reaches the detector from where you see it in the video scan. Okay, and on the TEM, you have an electron gun, you have the condenser lens, the specimen is placed much above, you have the objective lens over here, the projectile lens, and then it's directly viewing in a digital camera. So more or less they have they're used for different applications, but and are set up differently. So one is the scanning electron microscope, and another one is the transmission electron microscope. Right, so from the ground state you reach up to the excited state, and from the excited state to the ground state. So first one is absorption, and then due to emission you see actually that part. So you have a light source over here, it reaches over here, the object is placed, and then it directly reaches the eyepiece. So there are one or two barrier filters that are placed over here. The first barrier filter is over here that lets only the blue light a certain wavelength to pass through, and then the second barrier cuts out the unwanted fluorescent signals, allowing the specific emitted light, which is a green light, say, to reach up to there. And then the beam splitting mirror reflects the light below a certain wavelength and transmits light above that wavelength. So the confocal microscope is based on such a principle. So it has a pinhole that focuses the illumination at a particular point, and then you have another pinhole that collects the emitted light only from a point. Then you have the noise from out of focus points in the specimen is excluded. You have the fluorescent specimen, which is illuminated with a focus point of light from a pinhole. Then the detectors, where the emission fluorescent light from in focus point is focused at pinhole and reaches the detector. Now, emitted light is out of focus point, is out of focus at pinhole, is largely excluded from the detector. That is why focusing it is quite important. You see, it's a focal and this is a non-focal image. It depends entirely on the focus. Okay. Now, if we talk about the cytoskeleton, okay, so cytoskeleton is mainly important for support, motility, and the regulation. So, cytoskeleton helps maintain the correct shape and proper internal structures of the cells. It interacts with motor proteins to assist in motility. It helps the cells to change shape. Okay, to change the shape and move from one place to another. So during mitosis, it pulls the chromosomes apart and helps in the cell division. So there are three types of protein fibers that constitute the cytoskeleton. You have the microfilaments that determines the cell's shape and driven whole cell locomotion. Microtubules that determines the position of cellular organelles and direct intracellular transport. Intermediate filaments provide mechanical strength. So there are three types of protein fibers that constitute the cytoskeleton. You have the microfilaments, the microtubules, the intermediate filaments. So you have the actin, the alpha beta tubulin dimer, and then the various other types. Okay, so they have a structure. Then there are different subunits that are basically playing a role in the formation of the structures, the localization. For localization, there are different ways of how they actually function. So actin exists as a globular monomeric protein known as the G-actin and as its filamentous polymer called F-actin, which is a linear chain of G-actin. Now in the filaments, the subunits are organized as a tightly wound helix. Now all subunits in an actin filament point towards the same end of the filament. Consequently, a filament exhibits polarity, and that is one end differs from the other. So the actin filaments polymerization at the leading edge pushes. So continuous actin nucleation at the plus end facing the membrane. So there are branching structures that push the plasma membrane forward, and that is lamellipodium forward formation. Now the plus ends are protected from polymerizing by the capping proteins such that they do not form the polymers. The minus end near the center of the cell disassembles through the depolymerization. The web of actin as a whole thereby undergoes a continual rearward movement due to the assembly of filaments at the front and their disassembly at the rear. Okay, now the microtubule subunits as the alpha and the beta tubulins. So that forms the microtubule subunit. So even they have a plus end and a minus end. So the plus end is because of the beta unit, and the minus end is the alpha unit, and they form circular like microtubules like structures, and they can form it. So it's more or less like the tubulins. So microtubule associated with the motor proteins are the kinesins and the dyneins. So you have the kinesin is basically it has a head, the neck with the linker, then the stalk region where you have the heavy chains and the flexible hinges that are intertwined, and the light chains forms the tail. And on the other hand, the dyneins have basically the stalk, you have the heads, the heavy chains that forms the head, you have the stems that attach the intermediate and the lighter chains. So kinesin and dynein mediated transport of vesicles and the organelles. So our genome is highly compacted into the chromosomes. Okay, we know that. So you have the nucleus, you have the interphase chromosome, then it coils up to form the higher order chromatin folding. These are the loops, and then they coil around the histone proteins. So these are the like the beads of string. You have the nucleosome, and finally it forms the DNA. So basically DNA is wrapped around the nucleosome known as the histone protein. So you have H2A, H2B, H3, H4, things like that. So that's about the basics of the cell and things like that. Okay, so all right. So if you have any questions up to here, you can ask me. No questions? Okay. So now we will talk about cell division. So cell cycle has two distinct phases where you have the chromosome duplication and so which has you have the G2 phase, then you have the G1 phase. Okay, so that's so this is the mitotic phase. From two you go on to get the sister chromatids, and then you have the chromosome duplication. So what's the important event of this phase? So we will learn about that. So what is the significant about DNA in the S and the G2 phases? So all chromosomes are duplicated. That means there are two copies of each chromosome. So what are the copies called? They are called the sister chromatids. But do they contain identical genes? Yes, but okay. So what about the meiosis? So some terminology like we have chromatin, the homologous chromosomes, you have the chromatids, okay, double chromatid chromosomes, single chromatid chromosomes. So when do each of these occur? So we will get to know one by one. Now the steps of mitosis includes the interphase, prophase, metaphase, anaphase, and the telophase. So though interphase is not actually the part of mitosis, so in interphase, you have a nuclear envelope that's intact. The chromosomes are duplicated but not apparent. DNA loose, uncondensed, called the chromatin. Two nuclearly visible in onion root tip, and nuclei is not visible in whitefish blastula. Now the prophase is basically it's a double chromatid chromosome evident. Now chromatin becomes supercoiled and compact. You have a nuclear envelope that breaks down. Now the centrioles migrate towards the poles of the cell, forming the spindle. Now the double chromatid chromosomes looks like it. So you have one chromosome and its sister chromatid. The centromere is basically the center part of it. Now in the metaphase, the double chromatid chromosomes line up on equatorial plate of the cell. The spindle fibers attach to the chromosome at the centromeres. In the anaphase, things the spindle fibers pull the chromatids apart, the centromeres are pulled apart, they separate out. The groups of the single chromatid chromosomes move opposite poles of the cell. And in the telophase, there are groups of single chromatids that reach the poles of the cell. The nuclear envelope begins to reform. Two new daughter cells are formed, and cytokinesis begins with appearance of the cell plate and the nucleoli reforms. And on the other hand, the cytokinesis begins with appearance of the cleavage furrow. Now returning to interphase, so you have the cytokinesis that completes, then the chromosomes disappear as the interphase chromatin reforms. You have the two new daughter cells that enters the G1 phase of the cell cycle. So okay, so a brief summary of mitosis. So what happens? You have the interphase, then there is an early mitotic or the prophase. You have the late mitotic prophase, then mitotic metaphase, anaphase, and the telophase. So you see over here you have the centrioles is the chromatin, and then they start separating out, and two new daughter cells are formed. So what are the types of body cells called? So they are called the somatic cells. These cells divide continuously. The new cells receive an extract copy of the plant cells, and the process of somatic cells duplication is called meiosis. So what are the somatic cells? So these are diploid in number or the 2n. So what does this mean? That they contain the full number of chromosomes that is in pairs. So how many in humans? You have 46, that's 23 pairs. So each of these chromosomes, each pair of these chromosomes have a different function. They carry the different types of genes, some for the height, some for determining the sex of an organism, some for the different other functions, and they look also quite different. Now, mitosis occurs in somatic cells. So the sex cells are called the gametes. So gametes are not diploid or 2n. Instead, they are haploid. That is N. So our haploid number is 23. So our eggs and sperms have how many chromosomes? That's half the number. So fertilization is basically the union of an egg and the sperm. Now, if the egg and the sperm were both diploid, what would be the egg zygote be? It would be a genetic mess. So that is why egg and sperm both have exactly half the number of chromosomes of somatic cells, such that at mitosis you have the DNA replication, and you have the two daughter cells. Now, in meiosis, there is the parent cell, the DNA replicates, the two daughter cells are formed, and then each of the two, one from the mother and the father, you get the four daughter cells. So at fertilization, you have n. So one comes in the form of sperm, one in the form of an egg, and then that gives you 23 plus 23, that's 46. So otherwise, yeah, it would be quite difficult. Okay, so I don't know why I was like removed from this meeting, but that's fine. I'm back. Okay, so we were talking about right. So let's start from yeah. So fertilization is basically the union of an egg and the sperm. Now, if the egg and the sperm were both diploid, what would be the fertilized zygote be? So you would have 2n plus 2n, that's the 4n, and that would have been a complete mess because that would actually affect the entire thing. Because after that, it would be 4n plus 4n, that's 8n, and then oh, that's a complete haphazard way of doing it. So instead, gametes are haploid. That is, one cell comes from the sperm and one cell comes from the ovum, and then it's 23 plus 23 forming 46. So note how mitosis and meiosis differ. So yeah, you can see over here in mitosis you have the two daughter cells, and that's an haploid number. But for meiosis you have that's a diploid number for mitosis, and for meiosis you see a haploid number. For mitosis, you get two daughter cells, and from meiosis, you have four. So, meiosis somehow somatic cells in our ovaries and testes must produce the gametes N. So, meiosis is the cell division process that enables the transformation from 2N to N. That's an haploid number. Now, how is meiosis more complicated than mitosis? So, the gametes must contain precisely half the diploid number of chromosomes. They must contain one of each homologous pair of chromosomes. So if you remember the karyotypes, now each chromosome has a partner. They come in pairs, one from the mom in the form of ovum and one from the dad in the form of sperm. Now that's the human karyotype, and every sister chromatid looks different. The pair looks quite different, and every pair has a particular function. One for the height, one from the hair color, the eye color. Then it determines how tall you are. It will determine the sex chromosomes, which is the 23rd. That's X and Y. So if it's an X and a X, it's a girl, and if it's an X and a Y, it's a boy. So which pair of chromosomes is not in homologous? It's the 23rd pair. That's in males. It's the XY pair, and for females it's XX. So it's fine. So what happens? What homologous means? So all our other pairs of the chromosomes are homologous. That is, they are same. So how many divisions does meiosis have? So you have an interphase, then there is a homologous chromosomes that are formed. You have meiosis, the daughter nuclei, and then from there you have another four. So from 2n you have n, and that are four. Right, so one pair of homologous to start, the DNA replicates. From meiosis you get 2n to n, and from meiosis 2 the chromatids separate into the four products. So what you understand is that meiosis is very important for reproduction, for the growth of the cells, and for mitosis it's mostly because of the maintaining the cells, maintaining the cellular homeostasis, going on to the normal functions of a cell. That's important for maintaining the healthy cells. But for reproduction, it's the meiosis. So, diploid parent cell, you have the two haploid cells with each chromosome still consisting of the two chromatids, and then the four haploid progeny cells. Now, in males, how many progeny are produced? It's typically four viable sperm are produced following each meiosis 2. Now, in females, it's just one viable ovum is produced plus three small polar bodies. So what does crossing over mean? So, and when does it occur? It only occurs during the prophase of meiosis. Once homologous chromosomes get together into temporary tetrads, so the two chromosomes comes together and there is exchange over. So overlap, crossover, and they trade their DNA. Why is this a generally good thing to do? Because it actually maintains, it actually gives you a different type of a gene, and also it's important for having some kinds of different types of organisms. Otherwise, all the people around us would be basically the same if that have been maintained. So every person is different. They have different traits, characteristic features, things like that. So everything comes because of this crossing over, things like that. So meiosis one, it's the place where the DNA replication takes place. A parent cell produces two daughter cells, each with one member of each original pair of homologous chromosomes to create haploid daughter cells. So the crossing over may occur. Meiosis 2, there is no more DNA replication. The chromatids of each chromosome separate and each daughter cell divides. Now, at the end of meiosis 2, there are four daughter cells from each parent cell. Now, each daughter cell has half the number of chromosomes as the parent cell. Okay, yeah, so that's all from the lecture part. Any questions if you have? So we will start with the question and answers in like 2 minutes. Okay, so let's start with the questions then. Now, the largest cell on Earth is dash, and the smallest cell is dash. Their size is dash and dash respectively. So you have to identify the cells and the size. What is your answer? Yes. Yes, it is A. It's the ostrich egg, which is of 15 centimeter, and the Mycoplasma galactipsium, that's 0.3 micron. Yes, that's correct. The second one says choose the correct option about prokaryotic cells. So, yeah, so which of them actually fits with the prokaryotic cells? Okay, we got one answer. So prokaryotic cells, they lack a membrane enclosed nucleus and or other membrane bound organelles. They have a single circular chromosome. Their genetic material is not associated with any histone proteins, and the cell wall is primarily composed of the peptidoglycan. So so 3 and 4 are basically correct. So option C. Now coming to the next one, which is the correct option about the eukaryotic cells. What is correct about the eukaryotic cells? Okay, yes, that is correct. All the four options are correct over there. So they have a well-defined nuclei that house the genetic material, have membrane-bound organelles, they have histone proteins, and they don't have a cell wall, but plant and fungal cells have cellulose and chitin in the cell wall. Yes. The next one says it's match the following. So you have to match them with their functions. So you have the organelles and you have to match them with their functions. So you have the nucleus, mitochondria, lysozymes, and the smooth endoplasmic reticulum. Okay, so I have got one answer. Okay, so one is nucleus, so that contains the genome, DNA, and RNA synthesis. So yes, the mitochondria we know is the powerhouse of the cell. So that's option three. It's ATP synthesis by oxidative phosphorylations. Lysosomes, they basically lead to intercellular degradation, and the ACR for lipid synthesis. So option C, correct. Okay. Now the next one, fill the boxes mentioned in the figure provided. Okay, so basically, no. This questions, the answer is basically given entirely. So yeah, you can skip this. So this is basically the actual structure which I've put up. Uh, the boxes are basically missing from these spots, so you can skip it. Okay, let's go to number 6. Yeah, does not matter if it's A or B because I haven't put up the boxes, so it's quite difficult to tell. Okay. So which instrument is more useful to study surface characteristics of a specimen? So if you have to study the surface characteristics of a specimen, what would you use? A phase contrast microscope, a scanning electron microscope, a light microscope, or a transmission electron microscope? Yes, so the correct answer is a scanning electron microscope. It's designed to produce the specimen surface topography. So it will detect by scanning a focused electron being over the surface, and then the secondary electrons that are emitted to get the surface characteristics. That's correct. Now, induced pluripotent stem cells are created by. So how are induced pluripotent stem cells created? Yes, so induced pluripotent stem cells are derived from the non-pluripotent somatic cells. Now, this process involves the introduction of specific transcription factors, which converts the somatic cells into the stem cells state. Now, these factors that induce the cells to become pluripotent, meaning that they can make any kinds of cells. So, yes, it's reprogramming the somatic cells into embryonic-like state by adding some protein. So, it's option B. Yes. The next one is about the cytoskeleton. So, which of the following options are correct for cytoskeleton? C, yes, it maintains the correct shape and proper internal structure of cells. That's correct. It interacts with motor proteins to assist in motility, quite important. So, yeah, correct. It helps to change the shape and move from one place to another. So, it's associated with the motor protein. So, that's also true. And during mitosis, it helps in proper segregation of the chromosomes. That is also correct. So, option 8, that's C. Okay. Now the ninth question says in most eukaryotes are dash and the sex cells are dash. So quite easy. Okay, I've got one answer. So sex cells are generated after the meiosis cycle. So you know it's haploid. So yes, so and the eukaryotes are diploid. So most eukaryotes are diploid and the sex cells are haploid. Okay, so yes, that's option A. So diploid and haploid, not option C. Okay, and match the following where you have to understand the cancer terminology with their characteristics. You have leukemia, hyperplasia, malignant tumor, then metastasis, and the benign tumor. Benign tumor. Okay, so let's see. So what is leukemia? Okay, so leukemia is a type of cancer that affects the blood and the bone marrow, right? So it's 4, 1 gives me 4. Hyperplasia is an increase in the number of cells in a tissue or organ, often a precancerous condition, but the cells arrangement is normal. So that's 3. 2 comes to 3. 3 is the malignant tumor. It's cancerous growths that invade the nearby tissues and can spread to the distinct parts of the body. That's the metasize. Okay, and you have the metastasis, which is a process by which cancer spreads from an original tumor site to new tumor elsewhere. And the benign tumor, it's non-cancerous growths that do not invade the surrounding tissue or spread to the other parts of the body. So 1, 4, 2, 3, 3 comes to 2, 4 to 1, and 5 to 5. So that's option A. Yeah, fine. That's all. If you have any questions, you can ask me. If not, we're done for the day. If you have any questions from the previous classes, any questions, any doubts, any questions with the assignments, things like that, anything? No? Okay then, fine. Thank you for joining. We will meet on this coming Friday again. Okay? Yeah. Thanks.
Thank you, sir.